Yeah, it's a pleasure to introduce Michael. And Michael is the current third year bioinformatics PhD student in my lab. And he has been in our lab since last spring, 1.5 years already. Yeah, Michael graduated with a bachelor's degree in molecular biology and also in bioinformatics minor at UCLA. And so he has been here for a while. And in our lab, he mainly leads method development, mainly in the area of integrating multi-omics data from single-cell profiling and to understand the gene regulatory networks. Yeah, he is really technically strong and also very, very creative in developing a lot of the methodologies and also doing a lot of application work to understand various diseases. So today, Michael will mainly talk about one of the tools he is developing called scGRNdb. All right, Michael, the floor is yours.
Yes. Yeah, thank you, Dr. Yang, for the very kind introduction. Let me share my screen here. So, yeah, as Dr. Yang mentioned, one of the projects I'm focusing on and that I'm presenting on today is providing a cell type gene regulatory network resource for human and mouse for the public to use for their bioinformatics research. At the end of this talk, I hope to communicate the excitement of using gene regulatory networks to understand disease mechanisms, and also potential ways to perform this network analysis and integrate it with different types of omics data. So now I want to first motivate why, what is gene regulatory network? Essentially, it's a network of nodes and edges, nodes being the genes in your data set and edges being regulation between the genes. They're attempts to uncover gene interactions that comprise all the molecular regulation in cell type or tissue. Most of these GRN methods that have been created are optimized for bulk RNA sequencing or single cell RNA sequencing, though some methods are now integrating other omics types like single cell tag sequencing for chromatin accessibility and enhancer regions, as well as spatial transcriptomics to understand how regulation differs across the tissue. So reasons why we want to use gene regulatory networks to study disease is that it helps us identify key mechanisms in disease, mechanisms that are dysregulated between your case and your control. Understanding these mechanisms can help propose potential therapeutic targets or genes in the network that tend to be driving these disease mechanisms. And one of the most important reasons is to try understanding genetic variation in disease. Now, I want to connect how we can understand genetic variation from this transcriptomic data. And it really follows this model, known as the omnigenic disease model of genetic variation in disease. So the question that is longstanding in the bioinformatics research community is, how does genetic variation contribute to disease? This recent hypothesis or philosophy called the omnigenic disease model, posited by this paper here, Boyle et al. 2017 from Jonathan Pritchard's lab, describes it as any genetic variant regulating a gene in a tissue or a cell type has a small yet non-trivial effect on complex trait through these different cellular networks, networks that like gene regulatory networks that we've discussed already. There are some implications of this hypothesis. First, these genetic variants or SNPs that you would see in like genome-wide association study for a complex trait. Those with the largest effect sizes that pass genome-wide significance, they tend to regulate core disease-related genes and pathways, but this only explains a small fraction of this disease heritability. If you want to broaden the scope of the genes that could be affecting disease, maybe indirectly, not as these core genes, but maybe SNPs that are regulating genes that affect these core genes, that we can visualize in a network, right, of peripheral genes being connected indirectly to core genes. SNPs that regulate these peripheral genes can explain even more heritability. And these would be SNPs that don't pass your genome-wide significance threshold, or just very common variants that are involved in pathways or processes that may not be directly related to the disease. And furthermore, these SNPs can have regulatory effects that are specific and distinct to tissues and cell types. This small graphic here from the paper shows that each tissue has different gene expression profile, yet the same SNPs that are created from the genome-wide association studies can show different implications on these genes. And this does seem to make intuitive sense as well, given the heterogeneity of biological systems. And so with all this information, and understanding that genes can, peripheral genes can also affect core genes and influence complex traits, this highlights the need for GRNs, or gene regulatory networks, to understand tissue and cell type genetic mechanisms in disease. There are quite a few ways to build these gene regulatory networks. Many of the existing methods use different statistical frameworks or machine learning frameworks, like Bayesian inference, building a Bayesian network, directed acyclic graph of regulated genes to target genes, regression trees like random forest or gradient boosting, to try to capture the nonlinear interactions between genes, and also more associative metrics like mutual information or correlation. But in terms of the biological scope of these gene regulatory networks, they mainly split into two classes. The first being these knowledge-based gene regulatory networks. These ones restrict their features or their regulator genes in the network to known regulatory genes. For bulk RNA sequencing or in single-cell RNA sequencing, you can imagine these features would be like transcription factors with known target genes. For single-cell RNA sequencing, there also provides opportunity to understand ligand receptor interactions between cell types. And in this case, this knowledge base would be ligand and receptor interactions. The advantage of using these is that you're finding enrichment of known biology, which will give you very strong networks that explain true biology. But you do tend to overlook novel interactions that are not captured in these knowledge bases, or that are a completely different scope than the knowledge base you're looking at. For example, for transcription factors, they don't capture perhaps long non-coding RNA regulation of gene expression. For a ligand receptor databases, they may not capture different types of metabolite-mediated signaling. So one way to try capturing more modes of regulation, or trying to survey all of the interactions in your cell type or tissue, is to build these unsupervised gene regulatory networks. These ones would employ empirical feature selection methods across the entire gene space to look for potential regulators for each gene. Certain these types of feature selection could be running a pairwise correlation of genes, and limiting your predictive model to genes that have significant correlation with your target gene. Now, these can capture novel interactions, right, as we were broadening the scope of the genes we're looking at. But this does risk having a high false positive rate in your networks. These methods can tend to yield very highly dense networks, because many of the edges can be confounded by the features you're selecting. So there is a need to finding a balance between these two types of classes of GRNs, having a broad survey of potential regulation in your system, while also mitigating the false edges that can be introduced to the network. So this is the type of method that we've been trying to, that we have built for single-cell RNA sequencing, which I'll get into a bit later. But I want to first introduce, the revolution of single-cell RNA sequencing, as we know that we can now profile gene expression of individual cells and compare the gene expression, distribution, or patterns across cell types. But it does bring an important experimental constraint, which is high gene sparsity in your data set, where many genes have zero count due to poor experimental capture, but also just gene stochasticity or randomness in gene expression from cell to cell. And so that does tend to obscure the signal that is there in a cell type. So the methods that we develop, like for gene regulatory network methods, they have to take these things into account. So yeah, now I want to talk about gene regulatory network inference, now combining it in the era of single-cell genomics. There are many data atlases for single-cell data or to deposit other types of omics data. And they are only rapidly expanding. Like the human cell atlas, for example, right now has profiled over 50 million cells across a variety of human tissues, across 670 labs. And again, these numbers are only growing. And the resolution of the throughput of these single cell profiles are also getting better. It does highlight a critical need for robust GRN methods, right, to handle the experimental constraints of single cell RNA sequencing, but also comprehensive GRN resources for people to incorporate into their own studies, single cell studies. So we developed a single cell gene regulatory network inference method called scGRN, which employs gradient boosting approach and an unsupervised feature selection approach to build these robust gene regulatory networks. And then we're with scGRN, we're also scaling this algorithm to many different publicly available single cell data atlases. And we're curating it into what we call scGRNdb, or single cell GRN database, which currently is a cell type GRN atlas for human and mouse across 12 atlas repositories. So first I'll talk about this method before getting into the application of it. So the first thing scGRN does to optimize or accommodate single cell RNA-seq is performs a pseudo bulking of cells in the data set. So the input to this method would just be single cell RNA-seq with the cell types already labeled. And with your cell type, with each individual cell type, you perform a subclustering using a standard clustering method like leading clustering to generate these a number of subclusters within your cell type. And for each subcluster, we average gene expression to introduce these pseudobulk cells. This does help mitigate sparsity. It bolsters the zero counts in some cells in your subcluster back to like a more reasonable number based on the cells in your subcluster that do have expression for that gene. And then after this pseudo bulking, we perform a bootstrapping with 100 random subsamples, where each subsample would have 70% of the supercells or the pseudobulk cells in your data set and their associated average gene expression profiles. This way we build a network on each of these subsamples and find consistent trends or patterns between them. For the actual method of generating the networks, we use gradient boosting, which is a tree-based machine learning algorithm. Essentially, it builds very shallow decision trees on your data. And with each additional tree you're building, you're training it on the mistakes of previous trees. And as you iteratively add each contributing tree, the accuracy of your predicted value gets better and better. It more closely approximates your target variable. So we perform this gradient boosting for every single gene in our data set. Again, we are limiting the number of features, parent genes or regulator genes to predict each target gene expression using a k-nearest neighbor approach on the principal component space. We're taking about 100 nearest neighbor genes in the cell principal component space to reduce model complexity when building these networks. Again, we've built it on every bootstrap sample. And because of these bootstrap samples, there are slight variations between the networks. Certain edges that are present in some, but not others. And so one way to mitigate the false positives in our network is to find robust edges that are consistent across these networks. So scGRN applies multiple rounds of filtering. First is taking the edges with the highest feature importance as determined by each gradient boosting model. And then filtering out edges that are only unique to less than 20% of the networks, or the bootstrap networks. And then we prune out any type of confounding edges like triads, where two genes, there's edges between two genes, but also a shared upstream gene with those two shared genes. Something that looks kind of like this. We perform a conditional mutual information, as if you condition on the parent gene, what is the mutual information between the two target genes. If there is significant mutual information, then we would tend to keep that edge. But if there isn't significant mutual information, then we can take this edge out. So these are, the main filtering steps for scGRN. And again, reducing false positives would help bring to the front biological signal in these networks. And I'll get into how we can actually analyze these networks with like gene module detection, pathway annotation, and more guided disease study. Yeah, so here's where the single cell GRNdb comes in. Currently, we have curated 12 Atlas repositories. They have different single cell sequencing platforms, but within each cell type network, we're building it on one platform so as to not to handle any batch effects between data sets. And across these 12 atlases, we've profiled over 1,000 cell type gene regulatory networks across 90 tissues. Here I show just the top 20 tissues by number of cell type networks in them. We see that most of these networks have around 15,000 nodes and 50,000 edges. This is just brief numbers before getting into how we're actually interpreting these networks. So our plan with scGRNdb is not just to host these cell type networks, but also to provide analytical tools. One of these types of tools is understanding the network topology. So one way we can do this is by what we call module annotation or community detection within a network. Essentially, with our final network from scGRN, we can perform another leading clustering to identify highly connected subnetworks. And with these subnetworks, we can perform enrichment analysis comparing them to known gene sets like pathway genes or disease genes, and identify if there's an enrichment in the module for disease genes. When we do this, this helps us annotate or find biological significance to the topology of our networks. Here's an example of one analysis we did with a mouse primary motor cortex excitatory neuron network. Here I have the module number, the number of genes in the module, as well as enriched pathways and diseases. We can appreciate that there are very distinct physiological roles with these different modules, and they are related to the function of the neuron as well. Module 7, for example, has an enrichment of genes involved in neuron and synapse development, and has disease gene enrichment for a variety of neurocognitive related traits. We also see calcineurin-mediated signaling and regulation of vesicle-mediated transport in synapses. Again, providing confirmation that our modules are the topology of our network has biological meaning and significance. Another type of network analysis we are implementing is what is called key driver analysis. So the enrichment analysis I just mentioned, again, surveys network topology. But here we are performing a guided study. We're informing the network with genes of interest and wanting to identify where are the enriched hubs within our network, and are there any hubs within these networks that are driving the expression of these genes, or being aggregately affected by these genes. We call this key driver analysis. So what it starts with is first identifying these highly dense areas in our network. We would call these hubs. These hubs have very high node degree. And within each hub, we calculate the node degree of each direct edge to a hub gene, and sum up the weighted degree of each node. This helps us determine like the strength of the node the strength of the interaction of this node. When we now overlay the disease genes onto our hubs, we want to see if there's an enrichment of these disease genes in our hubs and if they're associated with these higher strength nodes. In a way, would be having a stronger connection or interaction within the hub. For this, we perform an enrichment analysis that's similar to chi-squared. Here we would make the observed measure the node strength or the ratio of the node strength of disease genes relative to the total, the sum of all of these node strengths. And then the expected would just be the expected node strength for any random set of genes that same size as the disease gene set. We would permute the network many times to generate a node enrichment distribution. And as in with any permutation test, see where our true data lies on this node distribution to decide if this hub is significantly enriched for these disease genes. And if it is, then the key drivers now we would term as the hub genes within these statistically significant disease-enriched hubs. Again, this would help determine for us what genes are termed key regulators in this disease of study. So here I'm showing an example of how to use these cell type gene regulatory networks for key driver analysis. I've queried all of the cardiovascular disease genes annotated by DisGeneNet, and I've overlaid them onto cinch gene regulatory networks to reveal these key driver genes here. The nodes that are in blue and that are larger are the key drivers and that are associated with cardiovascular disease. The ones that are in gray are not annotated as cardiovascular disease gene. Here we're looking at a liver hepatocyte network. And what we can appreciate is that a lot of these key drivers are related, are very distinct to hepatocyte function. Like these 3 genes here are subunits for fibrinogen, which is a blood clotting factor. And we know these to be associated with cardiovascular disease. And it's important that they're primarily produced in the hepatocyte, right? Another gene is this ApoB gene. ApoB is a component of many lipoproteins, which affects blood lipid concentration. Again, hepatocytes have a key role in uptaking and disposing ApoB. And then our networks can also propose these cell type specific genes that have no prior association to cardiovascular disease as annotated by an existing database, like this AGXT, for example. It has been shown to be dysregulated in non-alcoholic fatty liver disease, which also is quite correlated with cardiovascular disease incidence as well. Now, I also wanted to look at what are the key, using the same cardiovascular disease gene set, how do the key drivers differ in another cell type network within the liver? Here we're looking at the macrophage, which have clear functional difference between hepatocytes, and that macrophage is involved with more inflammatory processes. And we can see that these key driver genes are related to macrophage function, like IL-1B, which promotes inflammation, secreted by macrophages, the ApoE, which is secreted for lipid homeostasis, and then again, also proposing potentially new genes of study for cardiovascular disease, like this LITAF gene, which has been shown to be associated in non-alcoholic steatohepatitis, NASH. Sorry, I forget the acronym. But this causes secretion of TNF-alpha and other inflammatory mediators. And so what we can appreciate between this network and the network I showed before is that there are these cell type specific key driver genes that are related to cardiovascular disease and explaining different contributions of to this disease. So in summary, I just wanted to recap that we've built a single cell gene regulatory network method, cinch, and have applied it to single cell scGRNdb across many tissues and cell types across human and mouse atlases. And we're also providing statistical tools to analyze network topology and disease mechanisms. And in terms of future directions, we want to perform more systematic analyses of all of the networks we have. And connecting back to understanding genetic variation, we want to perform these network-driven genetic mechanisms using GWAS summary statistics. We'd also hope to incorporate different types of networks like co-expression networks into our atlas, and then we are actively developing a web server to host all these networks and analytical tools. We want to really tailor this to user needs, like providing a scG API for users to run scG instead of having to deal with the source code, or screening different networks from a gene set query to identify cell type networks of potential interest, and then also providing dynamic network visualization for others to study their diseases. Yeah, lastly, I just wanted to thank all the people involved in this work, like Dr. Yang, Russell, who was previous bioinformatics grad student in the lab, who helped develop, or who's the main author in developing scG, Jenny and Julie for working on the pipeline to build all these networks, and also for interpreting results, and then the rest of the Yang lab, again, for helping build all the networks across these atlases. So thank you for listening, and I'm open to any questions.